Hello, everyone, and welcome to today's webinar, Beyond the Noise, Identifying Clinically Relevant Mutations in Somatic NGS Testing. I'm Caitlin McCann-Jankovitz, and I'm the moderator for this on-demand event brought to you by QIAGEN Digital Insights. To learn more, visit digitalinsights.qiagen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask Question box and click Send. I'd like to now welcome our speaker, Dr. Anna Krivokuća. Anna Krivokuća is a clinical laboratory geneticist with more than 10 years of experience in clinical genetics and cancer research. She has implemented innovative genomic technologies into routine molecular diagnostics in Serbia, and she's experienced in the interpretation of genomic data, variant annotation, and genetic counseling. Dr. Krivokuća is the founder and the head of the Department for Genetic Counseling at the Institute for Oncology and Radiology of Serbia. She completed her Ph.D. studies in genetics from the School of Biology, University of Belgrade, and her master's studies in bioethics from Clarkson University and Mount Sinai Hospital, Department of Bioethics in New York. Besides her everyday work in clinical laboratory genetics, she's been actively working on many research projects and has been mentoring master's and Ph.D. students. Her research is focused mostly on genetics and molecular biomarkers in breast and ovarian cancers. She has published more than 40 scientific papers during her career and has been awarded numerous research grants and awards. One more thing before we get started, our legal disclaimer. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. For up-to-date licensing information and product-specific disclaimers, please visit www.qiagen.com. And with that, Dr. Krivokuća, I will pass it over to you to get us started.
Thank you, and thanks everyone for joining us today. So first of all, this is our outline for the webinar today. So first, I'm going to give you a short background of our institution, of the Institute for Oncology and Radiology of Serbia. After that, I'm going to speak about NGS in our lab, about how we deal with manual data curation, how we generate reports for our clinicians, and I'll give you some real-world experience and examples on how we deal with NGS data. After that, I'm going to show you a couple of examples of how we deal with complicated genetic reports that come to our department and our institution by outsourcing NGS data and NGS samples, and how to use genetic databases to adequately and reliably interpret the data that you have in front of you. Then I'm going to show you a couple of examples how to use genetic databases on molecular tumor boards and how clinicians can benefit from these databases. And at the end, I'm going to give you a sort of my personal suggestion how you can use these databases in order to generate your own pipeline for bioinformatics in your own setting, in your own lab. So I come from Serbia. This is a country in the Southeast Europe, and I live in Belgrade, which is the capital of Serbia. And my place of work is Institute for Oncology and Radiology of Serbia, which is primarily a hospital. So we have a large clinical infrastructure. We have a lot of patients that are referred from every part of the country. And we primarily deal with diagnostics and treatment of oncology patients. However, besides this, we are also a National Cancer Research Center. So we have a large research department, which is fully equipped. And we have a lot of PhD students and senior researchers, and we have a lot of national and international research grants. Besides regular everyday clinical and research work, we also have a sector for clinical trials where novel and innovative treatments are applied in our institution. And today we are going to speak about molecular diagnostics and our department that deals with molecular diagnostics, and particularly about our sequencing services and about our NGS platforms. So we started with molecular genetics back in 2008. We started with targeted mutation detection. We started with RAS testing for colorectal cancer, and we continued through EGFR and BRAF. We still perform these targeted tests for pharmacogenomics of colorectal cancer, lung cancer, and metastatic melanoma. In 2016, this was a year that was really important to us because we started performing cfDNA testing for lung cancer patients, both at diagnosis and at progression. But this year was also important for us because we were equipped with the NGS machine and we started performing sequencing through next generation sequencing machines. And we started by implementing germline pipeline, but we also started with somatic pipeline. And now we deal with both panels for germline and somatic in our lab. So when we talk about our sequencing service, I personally like to divide it into pre and post NGS era. Before 2016, we performed Sanger sequencing, but as most of you know, it was slow, it was inefficient, it was really demanding, and we were not able to incorporate this as a routine test for our patients. It took like a month to sequence only one gene, and we did it for hereditary cancers, but mostly for research purposes, and back then we didn't have tests covered by the insurance. So we did it mostly through our research projects, and we performed targeted sequencing. After we received one Illumina MiSeq machine in 2016, we switched our approach and we started performing sequencing faster. We were able to incorporate these tests into our routine clinical diagnostics. Besides germline testing, we started performing somatic testing for our patients. We established genetic counseling department. We were able to perform fast and efficient sequencing, even though we are still limited in the only one machine that we have. These tests nowadays are covered by the insurance, but our patients are also able to pay for the test in our institution if they do not fulfill the criteria for testing. In the meantime, we received accreditation for European Molecular Quality Control Network, and we also have our research publications where you can find our research projects that we perform here. I want to point out that we are actually a reference center for sequencing and that we get referrals for germline and somatic sequencing on the NGS machine from all over the Serbia. Also, we get the referrals for sequencing from neighboring countries. We have been establishing bioinformatics pipeline for almost 5 years now, and I have to admit that we are still evolving. But the thing is, since we come from a developing country and we have a couple of pipelines for NGS, you are not able always to buy, and you don't have the funding for these expensive softwares that is going to help you to perform variant analysis. So we established our own in-house pipeline, which is based on manual data curation. So we generate the reports, and we actually talk to our clinicians and generate the reports and do it in our own lab. So I'm going to give you an overview of how do we do it, what databases do we use, and I'm going to show you how a QIAGEN database, which is called Human Somatic Mutation Database, can help you and helped us in some of these examples. Okay, so for all of you who perform sequencing, you know that no matter what type of panel do you have, no matter whether you use smaller or large panel or even exome or whole tumor genome sequencing, it is always difficult to interpret the variations that you have in front of you. Sometimes you get stuck with just one. So it doesn't matter whether you have smaller or larger panel, the challenges with variant classifications are almost always the same. So you have a lot of information, you have the vast amount of NGS data, and the thing that you have to do for your clinicians and for the patients is to adequately interpret the clinical significance of these DNA variations that you have in front of you and that you detected by the NGS. So what do you have to do? You have to analyze and access up-to-date information of these variants. So you have to basically look through the information online to see whether their prevalence in healthy people is large or not. You have to see whether these variations are related to a disease of interest or not. You have to investigate a functional impact on the protein. You have to look for the results from the clinical trials. You have to do a lot of work to get to the information that you need and to find the evidence that you need to classify these variants according to some classification rules. And even though you have trained people, even though if you have a lot of experience in this, even if you know your way into the databases, sometimes this job can be really difficult. And we find ourselves stuck with just one variant that we are not sure how to classify. And this is because these data sources are numerous. There is a lot of information online. Sometimes they're heterogeneous. Some of these databases have the information that you need, some don't. They're quickly evolving, and sometimes they're conflicting. And that's why scientific circles have noticed that there have been a discrepancy between classification between laboratories, which is something that we must not allow, since each and every lab should clinically classify the same variant in the same tumor type in the same way in every part of the world. So what happened in 2015? In 2015, the first guidelines and common framework for variant classification was established. And it was established by American College of Medical Genetics and Genomics and Association for Molecular Pathology. And we still use those as a gold standard, as the main guidelines for variant annotation. Since 2015, a lot of guidelines actually showed up. And I've become aware of the newest one, which came out just a couple of weeks ago. But they all more or less do the same. So they tell you that you have to look for evidence online and to use the evidence to classify your variation in one of the classes that they suggest. So what do we do? No matter which guidelines you use, you have to classify the variations according to their pathogenicity, which means that you have to decide whether these variations that you have are benign or pathogenic. So we have 5 classes, class 1 being benign, class 2 being likely benign, class 4 and 5 being likely pathogenic and pathogenic. And the gray zone is class 3, which are variants of unknown significance, clinical significance. And sometimes it's easy. For example, if you have a nonsense or a frame shift mutation in your gene, which you know that is related to a disease, sometimes it's easy to classify the variation in class 4 or class 5. However, you have a lot of issues when you have variants that do not have information on them online, and when they are usually missense mutations that actually are not clinically actionable and are actually BUS. So what else do you have to do when you perform somatic mutation detection and when you perform mutational profiling of tumor, besides its pathogenicity or oncogenicity, as they call it nowadays? You have to look for a specific somatic mutation databases which will give you information about whether the mutation, the variant that you have in front of you, is actionable or not. What does it mean? So it means that you have to evaluate whether the patient who has the mutation that you have in front of you is actually eligible for targeted treatment. And what do? So you have to establish variant actionability status. So now here we have tier 1 to tier 4, tier 1 being variants of strong clinical significance, tier 2 variants of potential clinical significance, which might a little bit relate to class 4 and class 5 that I mentioned before. We have tier 4, which is benign or likely benign variant, and as class 3 or BUS, we still have variants of unknown clinical significance here, which has tier 3 classification. So in our lab, we perform a classification according to pathogenicity and actionability following mainly ACMG and AMP guidelines. And we have our own in-house pipeline, and this is how it looks like. The thing is that you have to maintain and keep your local database updated. If you have a good local database, which means that you have information of all mutations and variations that you detected in all of your samples, clinical samples, it's easier for you because every time you find a variation that is in your local database, you don't have to classify it again. So you already have it classified. You just have to check whether there is new information or it needs possibly reclassification. If you have a good local database, then you have a half of work done. So if you find a variant that is not in your local database, what do? what do? So we manually look through public mutation databases. So we assess variation databases, locus-specific, gene-specific databases. We perform literature search. We look through segregation studies that are published online. We look through functional studies, clinical trials, in silico data to reach the classification and to reach pathogenicity and actionability status. And when we decide on the status, we put the information in our local database and we make a full circle. So next time when we find that variation, we already have it classified. And this might seem like an easy job, but it's actually a really difficult job because it takes a lot of time to perform all the steps. It takes a lot of devotion. It takes a lot of knowledge, and you need trained people to do this. Because sometimes, even if you are aware how to do this, you still have conflicting data. You do not know which evidence to use, and you actually just go around in circles. So this can be really difficult. So I'm going to give you a couple of examples of how do we do it, and the examples are actually from our lab. So the first one, we had a breast cancer case, and we detected one missense mutation in BRCA1 gene. So it was missense substitution. So we never detected it before, so we didn't have it in our local database, and we wanted to classify it. First of all, we wanted to assess its pathogenicity. And we entered and we looked for the information in the gene-specific databases. So here, I'm just going to show you the pipeline and to show you how we reach the final decision. So in BRCA-share, which is a gene-specific database, it was shown that this was variant of unknown significance, and it has some evidence to back up this classification. And we put it like a potential VUS. Then we moved on to Leiden Open Variation Database to look for the evidence for its classification. And although most of the inputs here show that it is possibly class 3, we had two information that it could be likely pathogenic or pathogenic. So immediately we have difficulties to decide what to do. So what did we do next? We looked through other databases. We searched in clinical trial, in ClinVar, and in information available online. So they had the most updated information. It was from 2021, and we had information that it was reviewed by expert panel and that it was classified as pathogenic. So we now have 3 different classifications, and we have to decide which one are we going to use. So is it class 3? And if it is class 3, whether it's actionable or not. And another issue that we have, even if we decide that it's class 4 or class 5, likely pathogenic or pathogenic, are these missense mutations and substitutions actionable? So are these mutations that do not change the length of the protein biomarkers for targeted treatment? Because some of the drugs actually require truncated protein. This is the basic example. So what did we do? So what could we do if we miss proper classification? So if we decide that this variation is of uncertain significance, it's not clinically actionable, and this patient would not be a candidate for PARP inhibition treatment. So this is variant of unknown clinical significance, and this patient will not be treated with targeted treatment. In case we decide it's a likely pathogenic mutation or pathogenic mutation, it is clinically actionable, it has tier 1 or tier 2, then this patient is going to be treated with targeted treatment. So this is really important for you to realize that based on your classification in the clinical laboratory genetics, the decision and the choice of treatment is going to be made. So the clinicians need reliable information from the lab so they can do their job further on. So what did we do? We moved on to look for the evidence to back up our classification, and we tried in silico tools that actually investigate biophysical characteristics of amino acids changes together with the protein multiple sequence alignment. And we saw that this mutation happened that is probably disease causing because we found enough evidence to back up our classification. And we also searched through many in silico tools online to back it up, and we finally had our decision. So according to our classification, this variation is of likely pathogenic, it's of class 4. So according to ACMG guidelines, we combined all the evidence and we found 2 strong evidence of its pathogenicity. We found 2 moderate evidence of its pathogenicity and 1 supporting evidence of its pathogenicity, and we found none evidence favoring polymorphism. So our decision at this point was to classify it as a likely pathogenic class 4. However, if you remember, we need to investigate the somatic mutation databases. So we have to see first whether this mutation is actually available or published in somatic mutation databases. So we have to see whether it's published or seen or found in the disease of interest. So we look for breast cancer. We have to see whether there are clinical trials related and which investigated this particular mutation, and we need to evaluate whether this patient is actually eligible for targeted treatments. So we need to establish its actionability. So what did we do? We entered the genetic databases that are strictly related to somatic mutations, and we found little or no data, which is the worst thing that can happen when you have a little information about the variation that you have in front of you. So we tried the Cosmic and other databases, and it was not previously confirmed to be somatic. And we found no evidence about drug resistance or drug sensitivity here. It was only published in one breast cancer case, and this was a reference or publication from 2018. So we have no updated information about this variation. The thing is, now when you do not have anywhere else to look, you find yourself in a difficult position of not knowing how to classify or annotate this particular variant, and your clinicians actually need the information to decide on the further treatment. Then we realized that there is a new database available online, which is called Human Somatic Mutation Database. And we wanted to see whether there is enough information that will help with our classification in here. And here you see the main screen of the database. And here there are 2 tabs that I wanted to look further into, genes and alterations, to find whether the particular mutation that I want to investigate is actually in the database. I started with the genes tab and wanted to see what do they have available on BRCA1 mutations. So I was able to look through the alteration type or through the functional impact of the mutations on the protein or their actionability. So what can you do here? You can just click on the tier 1 or T2, these are that are clinically actionable, and see whether your mutation is inside. Or you can search by alteration type, and I did it. I wanted to see what missense mutations are actually actionable in BRCA1 and to look for this particular that we had. And you also have everything really user-friendly, and it's really transparent, and you can click wherever you want to get to the information. So what did I do? I find the mutation in the gene tab and in the BRCA1 missense list of mutations. And I found evidence that I needed to classify it as tier 1, which is actionable mutation. So there was a lot of information. And the thing is, you have links to the other databases and publications that you want to further investigate to back up your classification. So of course, I found information that I already had, but I found additional information that I couldn't find anywhere else. So it was a variation with the known inactivating functional impact. It is actionability status tier 1. And I found a lot of clinical trials related to this particular variant and drug association. And the source was QIAGEN knowledge base with 54 references, the newest one being from 2021, if I'm not wrong right now. But I found updated data and information that I could just click on and just reevaluate it myself. And this is how it looks like in the system. So up here, you see the basic information that if you do not want to go further into, it's more than enough for you. Then you have biochemical impact. So you don't have to go in each and every in silico tool separately. So you have it all in there. Then you have the information that I looked for, which is related to clinical trials and to the drugs. So I wanted to see which drugs are approved by and suggested by European Society of Medical Oncology for this particular mutation. And what else did I do? I looked for breast cancer clinical trials that are possibly could be interesting for our clinicians and are related to this particular mutation. So at the end, this mutation was likely pathogenic. It was actionable, tier 1 level of evidence, and it was more than 100 clinical trials available to investigate further related to these mutations, 34 for breast cancer, 15 phase 2, 4 phase 3. We found 86 drug associations and PARP inhibitors as targets, and more than 50, I think 54, 5 references to back up this classification. And here I want to point out that when you find yourself in a difficult position of not having enough information to classify your variation, you might easily miss important evidence if you do not know how to reach them, or it's just not available to you, or you have to pay for the software or for the database, and then you're going to completely miss the classification, which can have extremely negative impact to the patient care later on. Another example comes from ovarian cancer case that we had. So it is a BRCA2 mutation, which is not a missense mutation. It is a splice site mutation, and immediately it was a problem because there was little information about this mutation. So for pathogenicity, it wasn't that big a deal because it is a frame shift mutation, so it was easy to classify. But if you want to classify it for its actionability, you need the data to back it up. So it's not enough to say it's a frame shift mutation. You need clinical trials, you need evidence, you need the data. So I started with the somatic mutation databases, and I couldn't find it anywhere. So this is the mutation, but these are different genes, so it's not in BRCA2. And of course, again, difficulties to perform classification when the evidence is scarce. Then I knew that I had HSMD, and I started immediately with HSMD. I didn't want to lose any more time, and I find evidence enough for me to classify the variation. So this particular variation, you have the explanation. It's located at the conserved splice acceptor site, and it's probably resulting in the loss of the majority of the protein, and there is information about publications. And it's already observed in clinical cases, concretely here in prostatic adenocarcinoma. Only one case reported though, but it's still important information for the clinician if they want to think about off-label therapy or something else. It's a tier 1. It has reported 110 clinical trials related to it, 82 drug association information, and source guidance knowledge base with a lot of references that you have. So here I want to point out that sometimes when you know that you have a reliable database and then in which is transparent, so you can go whenever you want in the online from that database, it's easier for you, so you don't have to waste time looking to the separate databases or in silico tools when you know that you have everything embedded in one. And the third and final case for this part of the webinar is, again, ovarian cancer example case, and it is a BRCA1, and this example I'm particularly proud of. We detected this mutation back in 2018, so it was a deletion of one nucleotide in BRCA1. So it was, you see it here, it was near splice site, so it was exon 12. And immediately we knew that it's a frame shift mutation, so we confirmed it with Sanger sequencing. But the interesting part about this mutation is that it was never, ever published before. So we were the one to find it first, together with the colleagues from Slovenia who also had, interestingly, the same case. So it is the two cases of ovarian cancer with the same mutation. We published this mutation as being the first one to publish it. So we had a great collaboration back then. So we performed deeper analysis. We performed analysis on transcriptional level, and we assigned one very strong evidence of its pathogenicity, assigned 3 moderate evidence of its pathogenicity, and 1 supporting evidence. And we classified it together as class 4 and published it back then in 2018. So I did a sort of retrospective analysis in HSMD. Back then, it was really difficult to classify it and to look for the information because there was none. Now it's easier because it's already embedded in the HSMD. So there is a lot of information about this mutation. Some of them we already knew because we classified it before. But important thing is that there is clinical trials and information about drug association that we didn't have back then, and also a source that is QIAGEN knowledge base. And I wanted to see the publications. And the thing is, these are our publications that we published in 2018 and 2019. So here I want to point out that it is really important to have updated information, because a year or 2 in genetics nowadays really makes a difference. So reliable, transparent, and up to date. For the second part, let's say, of our webinar, I'm going to show you how to use databases to deal with complex genetic reports and complex molecular tumor profiling, let's say whole genome or whole exome of tumors. These we do not perform in our lab since we are limited with the throughput of our machine, but I hope that in the upcoming year we are going to be equipped with the modern and larger machine, so we will be able to perform these analyses in our lab as well. However, you all know that genetic testing has becoming more available to our patients, and clinicians are dealing with much larger amount of information and amount of genetic data than ever before. So we see patients and institutions outsourcing NGS for complex analyses and calling us with the information that are just too much and that we all need together to analyze, to come together to analyze. This is the example. So this was a lung cancer case, and this was just the part of the complicated report, which was written on 50 pages. And on your left, you see the genomic findings that were written as actionable. And on your right, you see a list of genes and variations that were classified as variants of unknown significance. So they left it in the report, saying that they choose to include them in the event that they become clinically meaningful in the future. So here we have 2 issues. First of all, these VUS, who is going to classify and reclassify them, and when? And another thing is whether these information and all of these mutations on your left are actually important for your lung cancer patient. So what do? We use databases to assess these variations and to find the information that our clinicians need. And our clinicians asks us to help them with the interpretation of particular variation. Here we wanted to see whether these 2, FGFR2 and JK1, are actionable and could be used for a treatment. And again, I used HSMD to find the information that we need. The first one was tier 3, the second one was tier 2, but both of these had the information about possible clinical trials and that should be considered. So the information that I found helped the clinicians actually direct them into the direction of browsing for clinical trials, even though the thing is there is not much information about the drugs that are related to these variations. Another thing that you can do in the database, you don't have to look for a mutation, particular mutation. You can look in the tab that is called diseases, and you can look, for example, for lung cancer and search for all alterations that are reported currently as important and actionable in lung cancer. So you can do it that way as well. So when you look for lung cancer, you can look for the variations that are actionable, and you can look for the particular one that you are interested in. So there is 2 ways, and both of them work fine. The third thing that you can do using HSMD, you can evaluate these secondary findings and search for new molecular targets. Because most of the time when you have complex reports from whole exome of the tumor, you have information that you didn't look for. So sometimes reports might come back with information you haven't been searching for. And these are common in the NGS era. And these are not called incidental findings anymore. They are called secondary findings. And sometimes, even if you didn't expect it, you can have important information which is hidden in the vast amount of genetic data. So you can use HSMD to evaluate these secondary findings and to establish their role in a disease and to evaluate their therapeutic potential and to find possible match for drugs or clinical trials. And also, as I mentioned to you, a list of VUS can be really, really long. It is questionable whether you are going to reevaluate them anytime soon or not, but if you do wish to reevaluate these variants of unknown significance periodically, and if you have a whole pipeline for managing these VUS, you can easily do it in HSMD. How can you use this database in molecular tumor boards? I think this is really useful for clinicians because in these molecular tumor boards and tumor boards, when you have patients in front of you and when you have a lot of information that you want to, this cannot have enough time. So you need a quick, fast, reliable, and efficient, if nothing, database that is going to help you and answer you quick queries that you have on reported and variations that you have in front of you. What can you do? And I think that for clinicians, these three tabs at the end on your right are more important than genes and alterations. So explore diseases, explore drugs, and explore trials. So when you have a genetic report, whether it comes from your own lab or it comes from a different lab, you can use HSMD to quickly find the information on the tumor genomic profile, on the variant that you have in front of you, to establish actionability in just a couple of clicks away, and to match clinical trials with a specific genomic profile of your patient. So it can be a quick aid for you when you have no time to discuss about complex genetic reports, but you need to make decisions on the treatment and clinical follow-up. So in the Explore Diseases, you can browse through the disease description, you can look through the disease of interest, and you can search for genes that are enriched in your disease. You can look for alterations, you can look for drugs, clinical trials, and bibliography. In the Drugs tab, you have a list of drugs. You have gene alterations response related to the drug of interest. You have a list of sources, you have novel information of label therapies, and in the Explore Trials tab, you can search for clinical trials by their ID, and I think the most important for developing countries is geographically. You can look for clinical trials which are geographically closer to you by country or by location. And this is just an example of what questions this database can answer for you. For example, if you have a patient with a specific tumor profile, you can quickly answer, is the patient eligible for targeted treatment based on the molecular profile, especially if you have a limited list of the drugs available in your country. So you know that for lung cancer, you have 5 or 10 drugs available. You can easily see whether the patient's tumor profile is actually a match for the drugs that you can prescribe them. Or you can consider an off-label therapy. You can also look for a clinical trial that can be considered for your patient, and you can find a location of the clinical trial. So you have everything in one place. And as sort of my suggestion of how to use these databases wisely is that you, no matter whether you have enough funds and whether you have many pipelines in your lab or you have softwares, sometimes every software is missing something that you need. So you have to combine many. It's a lot of money. But at the end, you end up manually curating data, no matter whether you use softwares or don't. So if you have your own bioinformatics pipeline, it's always best. but you have to relate and connect your database and your pipeline with the databases that are available online. So how can you do it? If you remember, I showed you our pipeline for data and variant annotation. So it is complicated. It takes a lot of time if you perform it manually. But if you have a database that can do it for you, or that can embed the information that you need in one place, it's going to be easier. So you can skip all the steps that I showed you here. So you can skip search through databases, through the literature, to segregation studies, clinical trials by yourself in many tabs in your browsers. You can find everything in the database. So you need to have your local database. You need to have online database, which is genetic database, which is reliable, which is transparent. So you can easily find and back up information that you have in the database to assess pathogenicity and actionability data. So I believe that having a database, it's really important step in your bioinformatics pipeline. And at the end, I just wanted to just recap and emphasize the clinical applications of HSMD here. So it can help you perform manual variant annotation. So if you do not have the information about the variant, it's the first one that you see it in your samples. So you can manually annotate it using this database. You can also reevaluate up-to-date information on variants. If you have your database, but you think you classified it like 5 years ago, you can just reevaluate the information and reclassify it if necessary. So you can use it to discriminate between the actionable variants and VUS, or reevaluate VUS timely. You can use it to analyze complex genetic reports from complex molecular tumor profiling and put them into clinical perspective and give your clinicians the information that they need to treat the patients further. You can also evaluate molecular profiles of complex diseases. So you can use the database to investigate the genetic profiles and molecular profiles of the disease that you have in front of you. You can also find a solution for these quick queries that you have on molecular tumor boards. This is, I think, it's important for clinicians. So it can function as an app. Basically, it's fast, it's quick. You can find easily whatever you want. So you don't have the time, you don't have to waste the time just browsing through the internet. And for the people who work in the lab, I think that this database can be and will be useful if they want to establish their own in-house bioinformatic pipeline, and if they perform manual data curation, it's easier for them to have the database that's just going to make the things faster and more reliable. And with that, I would want to thank you for your attention. Thank you all.
Thank you, Dr. Krivokuća, for your informative presentation and your very important work. This does conclude our session today. To the audience, questions submitted during this event will be addressed via the contact information you provided at the time of registration. Thank you again for joining us today. Take care, and we'll see you at our next session. Goodbye.